Dear students, welcome you all. Today we are going to discuss plasmids. What are plasmids? Plasmids are transferable extrachromosomal DNA molecules capable of autonomous replication. They are double-stranded, mostly circular, and usually occurs naturally in bacteria and sometimes in eukaryotic organisms, for example, Saccharomyces cerevisiae. The size of plasmid varies from 1 to over 400 kilobase pairs. There may be one copy for large plasmids to hundreds of copies of the same plasmid in a single cell, or even thousands of copies for certain artificial plasmids selected for high copy number, such as the PUC series of plasmids. Every plasmid contains at least one DNA sequence that serves as an origin of replication, which is a starting point for DNA replication. And DNA enables the plasmid to be duplicated independently from the chromosomal DNA. The plasmids of most bacteria are circular, but linear plasmids are also known, which superficially resemble the chromosomes of most eukaryotes. Frequently, plasmids contain some genes advantageous to the bacterial host, for example, resistance to antibiotics, production of antibiotics, and degradation of complex organic compounds. Antibiotics are antimicrobial agents that can be classified based on their mode of action. Bacterial antibiotic resistance genes encode proteins that inactivate these agents either by preventing their accumulation in the cell or by inactivating them once they have been imported. By utilizing these antibiotic-resistant genes as dominant genetic markers in plasmid cloning vectors, it is possible to select for E. coli cells that have maintained high copy replication of plasmid DNA molecules. Plasmids used in genetic engineering are called vectors. They are used to transfer genes from one organism to another and typically contain a genetic marker conferring a phenotype that can be selected for or against. Plasmids serve as important tool in genetics and biochemistry labs, where they are commonly used to multiply or express particular genes. Many plasmids are commercially available for such use. Another major use of plasmids is to make large amounts of proteins. In this case, researchers grow bacteria containing a plasmid harboring the gene of interest. Just as the bacteria produce proteins to confer its antibiotic resistance, it can also be induced to produce large amounts of proteins from the inserted gene. This is a cheap and easy way of mass producing a gene or the protein. It then codes for example, insulin or even antibiotics. Some important plasmid vectors. Number 1, PBR322. This was one of the first artificial cloning vectors to be constructed and is undoubtedly the most widely used cloning vector till now. It is a 4.36 kb double-stranded cloning vector. This plasmid vector has been put together from fragments originating from 3 different naturally occurring plasmids. It contains call E1 origin of replication with relaxed replication control. Generally, there are 15 to 20 molecules present in a transformed E. coli cell. But this number can be amplified by incubating a log phase culture of col E1 carrying cells in the presence of colorum phenocol, like col E1, PBr322, and all its derivatives are amplifiable, which makes it very simple to isolate large amounts of these plasmids. PBr322 contains two antibiotic resistance genes. One is ampicillin resistance gene, coding for a beta-lactamase, which modifies ampicillin into a form that is non-toxic to E. coli. And the other is tetracycline resistance gene, which are set of genes coding for enzymes that detoxify tetracycline. The complete nucleotide sequence of PBr322 has been determined. The plasmid contains 20 unique recognition sites for restriction enzymes. 6 of these sites, that is EcoR5, BAMH1, SPH1, SAL1, EXMA3, and NRV1, are located within the genes coding for tetracycline resistance. 2 sites for HIND3 and CLA1 lie within the promoter of the tetracycline resistance gene, and 3 sites for PST1, PVU1, and SCA1 lie within the beta-lactamase gene. Cloning of a DNA fragment into any of these 11 sites results in the insertional inactivation of either one of the antibiotic resistance markers. The gene of interest is spliced into tetracycline resistant gene cluster, and then the E. coli cells are transformed. Thus, 3 types of cells are obtained. Number 1, cells that have not been transformed and so contain no plasmid molecules and will be ampicillin sensitive, tetracycline sensitive. Number 2, cells that have been transformed with PBr322 but without the inserted DNA fragment or gene will be ampicillin resistant, tetracycline resistant. Number 3, cells that contain a recombinant DNA molecule, that is, the DNA fragment has been inserted into the PBr322 at tetracycline resistant gene cluster. These cells will lose tetracycline resistance because the fragment has inserted in the middle of tetracycline resistant gene cluster. These are recombinants and will be ampicillin resistant, tetracycline sensitive. Now, cloning of foreign DNA in E. coli with PBr322. If foreign DNA is inserted at the PST1 restriction site, the ampicillin resistance element is disrupted and inactivated. After ligation of the DNA and transformation of E. coli cells, the cells are grown on agar plates containing tetracycline to select for those that have taken up a plasmid. Individual colonies from these agar plates are transferred using sterile toothpicks to matching positions within a grid on 2 additional plates. One plate contains tetracycline, a control, and the other contains both tetracycline and ampicillin. Those cells that grow in the presence of tetracycline but do not form colonies on the plate containing tetracycline plus ampicillin contain recombinant plasmid. As you already know that the ampicillin resistance element becomes non-functional with the insertion of foreign DNA. Cells that contain PBR322 that was ligated without the insertion of a foreign DNA fragment, retain ampicillin resistance, and grow on both plates. Identification of recombinant clones requires selection followed by screening. Now, number 2, vector PACYC184. This is a small 4 kb cloning vector developed from the naturally occurring plasmid P15A. It contains the genes for resistance to chloramphenicol and tetracycline. The chloramphenicol gene has a unique EcoR1 restriction site, and the tetracycline gene has a number of restriction sites as mentioned in PBR322. Insertional inactivation of one of the antibiotic-resistant genes facilitates the recognition of strains carrying plasmids with cloned DNA fragments. Like PBR322, it is not self-transmissible and can be amplified. It has been a very popular cloning vector since it is compatible with the COL-E1 derived plasmids of the PBR322 family and can therefore be used in complementation studies. Number 3 vector, PUC vector. The name PUC is derived because it was developed in the University of California by Messings and his colleagues. These plasmids are of 2.7 kb and possesses the COL-E1 origin of replication. These vectors contain ampicillin-resistant gene and a new gene called LAC-Z, which was derived from the LAC operon of E. coli that codes for beta-galactosidase enzyme. Number 4 is vector PUN121. This is a 4.4 kb vector derived from PBR322. This vector has been developed for rapid selection of bacteria containing a recombinant plasmid. This is required when genomic libraries are constructed, which involves the construction of several thousands of clones and hand-picking of individual clones with inserted DNA would be extremely tedious. Direct selection vectors have therefore been constructed. In these vectors, the original promoter region of the tetracycline resistance gene is exchanged for the PR promoter of bacteriophage. Transcription from this promoter is suppressed by the protein encoded by the lambda C1 gene, which was also cloned on PUN121. Cells harboring PUN121 will therefore have the phenotype ampicillin-resistant tetracycline sensitive as the C1 gene product suppresses expression of the tetracycline resistance gene. The C1 gene contains unique sites for the restriction enzymes EcoR1, HIND3, BEN, and SAMA1. Cloning of foreign DNA into these restriction sites will cause insertional inactivation of the C1 gene, abolishing the suppression of PR promoter. Cells harboring such a plasmid will become tetracycline resistant. Plating of transformed cells on the plates containing tetracycline will therefore only result in growth of cells harboring the PUN121 vector with inserted DNA fragments. PUN121 plasmid vector is a positive selection vector when no foreign DNA is inserted in one of the restriction sites in the C1 gene. The C1 gene product is synthesized and represses the PL promoter. The cells harboring such plasmids are sensitive to tetracycline. B. Plasmids with foreign DNA inserted in the C1 gene produce a truncated repressor. The PL promoter is depressed and the cells become tetracycline resistant. Yeast plasmid vector. Yeast is a single-celled eukaryotic microbe which can be cultured and manipulated using the standard techniques applied to bacteria. The genetic material of yeast is packaged into chromosomes within a membrane-enclosed nucleus and partitioned at cell division by mitosis and meiosis. In 1976, it was reported that a fragment of yeast DNA, when cloned into E. coli, restored histidine-independent growth to strains carrying the His-B mutation. Similarly, ligated fragments of yeast genome into plasmid called E1 and obtained a small number of leucine-independent transformants from a Leu-B E. coli strain. In each case, the yeast chromosomal segment carried the gene for the yeast enzyme equivalent to that absent in the bacterial strain. Thus, the yeast His3 gene was able to be expressed in a bacterial cell and produce the yeast version of the enzyme imidazole glycerol phosphate dehydratase and thereby complement the mutation in the bacterial His-B strain. Other yeast genes isolated in this way include Leu2, URAT3, TRP1 and ARG4. In general, genes from eukaryotic organisms rarely function in bacterial cells, but the success with yeast genes arises from the lack of introns in most such genes, coupled with the ability of some yeast promoters to function in bacteria. The wild-type yeast genes, thus isolated, provided essential markers in subsequent attempts to introduce exogenous DNA into yeast cells. Henin and co-workers in 1978 demonstrated that the yeast Leu2 gene carried by the plasmid called E1 could be taken up by Leu2 yeast cephaloblast and transform them to a leucine-independent phenotype. In this case, the transformation resulted from the integration of the added DNA into a yeast chromosome. Thus, a variety of approaches have resulted in a cloning system that makes use of many of the powerful techniques developed for E. coli. Now, what are yeast episomal plasmid? It is a yeast vector with the ability to replicate autonomously without integration into a yeast chromosome. Yeast vectors with this property have been built around a naturally occurring yeast plasmid. This molecule has no function and hence its name is based on contour length. It is present in most yeast strains with a copy number of 40 to 50 per haploid genome. The 2 micrometer plasmid is 6 kb in size. These are autonomous and show high frequency transformation. Yeast integrating plasmid. These are not actual yeast plasmid vectors because they are unable to replicate within a yeast cell without integration into a chromosome. Such vectors contain a yeast marker as the only addition to the bacterial plasmid. An important development in the use of these vectors has been the observation that opening the plasmid within the yeast segment before adding it to the yeast cells greatly increases the transformation frequency. For that purpose, the plasmid is treated with a restriction enzyme that has a unique recognition site within the yeast segment. if a plasmid carried two yeast segments, for example, URA3 and LEU2, cutting the plasmid within one of these regions will target the integration to that chromosomal locus. TI plasmids. Most cloning vectors for plants are based on the TI plasmid, which is not a natural plasmid but belongs to a soil bacterium, Agrobacterium tumefaciens. This bacterium invades plant tissues, causing a cancerous growth called a crown gall. During infection, a part of the TI plasmid called the tDNA is integrated into the plant chromosomal DNA. There are RI plasmids present in Agrobacterium rhizogenes, which cause hairy root disease in plants. Now, organization of TI plasmid. TI plasmids of Agrobacterium are large circular DNA molecules up to 200 kb in length, with molecular weights of about 3 to 8% of the Agrobacterium chromosome. They exist in the bacterial cells as independent replicating genetic units. TI plasmids have major regions for virulence, origin of replication, conjugation, oncogenicity, and catabolism of opines. Genetic organization of these regions has been shown in octopine TI plasmid DNA. Now, what are vector systems? Most yeast vectors developed share the following common features. Number one, most of the vectors are derived from bacterial plasmids and retain both the ability to replicate in bacteria and selection markers suitable for use in bacterial systems. As plasmid amplification is not high in yeast as compared to that in E. coli, this is a more useful feature. Thus, initial plasmid construction is done in E. coli before transfer to yeast. Such vectors permitting cloning in two different species are referred to as shuttle vectors. Number two, in most cases, the selection markers employed are nutritional markers such as LEU2 or HIS3. In some cases, such markers are not suitable. For example, brewing yeasts are polyploid and it is hence not possible to obtain auxotropic mutants. In such cases, resistance to copper or to the drug chloramphenicol have been used. Now, replication of plasmids. Most plasmid vectors in current use carry a replicon derived from the plasmid PMB1, which was originally isolated from a clinical specimen, and its close relative is called E1 replicon. Under normal conditions of growth, 15 to 20 copies of plasmids carrying this replicon are maintained in each bacterial cell. The PMB1 replicon does not require plasmid encoded functions for replication. Instead, it relies entirely on long-lived enzymes supplied by the host. Replication occurs unidirectionally from a specific origin and is primed by an RNA primer. Replication of plasmid DNA is carried out by a subset of enzymes used to replicate the bacterial chromosome. Usually, the plasmid contains only one origin of replication, together with its associated cis-acting control elements. The control of plasmid copy number resides in a region of the plasmid DNA that includes the origin of DNA replication. The F plasmids encode the gene required for bacterial conjugation. This extra chromosomal genetic element is approximately 95 kb and was named because of its role in bacterial fertility. The F plasmid is present as one copy per cell and encodes approximately 100 genes, some of which are required for F plasmid transfer during conjugation. As a result of integration into E. coli chromosome and then an imprecise excision resulting in the reformation of a circular plasmid, the F plasmid is able to transfer portions of the E. coli genome into another bacterium via conjugation. F plasmid containing E. coli strains are required in molecular cloning involving M13 filamentous bacteriophage. The F plasmid carries the sex pili gene, which encodes the cell surface binding site for M13 phages. Now, we will discuss some important features of cloning vectors. Number 1. Reduction in size of vector to a minimum to expand the capacity of vector to clone large fragments. Since the efficiency of transformation of bacterial cells drops drastically when plasmids larger than 50 kb are used, the size of cloning vector should be small, preferably 3 to 4 kb. In this way, foreign DNA fragments of 10 to 12 kb can be accommodated. Number 2. Introduction of selectable markers. Number 3. It should contain an origin of replication that operates in the organism into which the cloned DNA is to be introduced. Number 4. Introduction of synthetic cloning sites termed polylinker restriction site bank or polycloning sites that are recognized by restriction enzymes. This polycloning site is usually present inside a marker gene so that with the insertion of foreign DNA, it will inactivate that marker gene and the recombinants can be selected. Number 5. Incorporation of auxiliary sequences such as visual identification of recombinant clones by histochemical tests, generation of single-stranded DNA templates for DNA sequencing, transcription of foreign DNA sequences in vitro, direct selection of recombinant clones, and expression of large amounts of foreign proteins. Dear students, so far we have discussed nature, structure, types, importance, and replication of plasmids. Hope that you have understood the topic. Thank you.